Hello young guys, you are welcome in the classes of zoology. A few weeks ago we were talking about biological transport mechanism and intracellular trafficking part 1. Today in continuation of that we will talk about part 2. We have discussed about diffusion, in which there is movement of molecules or ions from their higher concentration to lower concentration. Then there is a facilitated diffusion, in which there is movement from higher concentration to lower concentration, but some carrier proteins participate in that. And then there is active transport. There also carrier proteins take part, but there the transfer of molecules or ions is from lower to higher concentration. So today we will talk about facilitated diffusion and active transport. Let us talk about facilitated diffusion. So in facilitated diffusion also there is movement of molecules or ions from their higher concentration to lower concentration. But here a carrier protein takes part, and this carrier protein have an active site or binding point for attachment of the molecule which has to be transported. So that molecule comes to attach with this binding point, and with this binding a conformational change takes place in the carrier protein and it rotates this way, comes here. Now this molecule detaches and go out. So it is a diffusion, but here there is a participation of carrier protein and their active site for the movement of molecules. Now comes active transport. Here in active transport also there is participation of a carrier protein, but here the movement of ions is from lower to higher concentration. It can be primary active transport, it can be secondary active transport. In primary active transport, the most popular example is about sodium potassium ATPase, which is the key point for development of resting potential in nerve fibers and muscle fibers. Here you can see that in this carrier protein there are three important points. First of all, on the inner side of the cell membrane, there are three active sites for 3 sodium ions. On the outer side, there are 2 active sites for binding of potassium ions. And the inner side of the carrier protein also contains ATPase activity, which breaks ATP into ATP phosphate and releases energy. So what happens? Here, just opposite to the concentration gradient, the sodium ions bind here, potassium ions bind here, and along with the binding of both ions, the site of ATPase becomes activated. It breaks an ATP molecule into ADP, phosphate and energy. That energy provides energy for movement of these ions and movement of the carrier protein. Now, this active transport may be of 2 types, primary active transport and secondary active transport. Primary active transport is where energy is supplied by ATP, GTP or CTP, and it causes the movement of a molecule. Then there is secondary active transport. You can see here that after the active transport of sodium ion and potassium ion, what will happen? Much more sodium ion will come on the outer surface. Then there will be a gradation in the concentration of sodium ions here and sodium ions here. So these sodium ions which are in higher concentration here, they will have a tendency to shift on the inside, and that tendency supports for the transport of some other molecules also along with it. So that is called secondary active transport. And the secondary active transport may be of two types. It can be co-transport or it can be counter-transport. Here you can see example of co-transport, where along with sodium ion there is one more active site for binding of glucose molecule. Both molecules bind, and along with binding of both molecules, immediately there is conformational change and the carrier protein shifts like this. So the sodium ion and glucose both are brought here. So here, along with movement of sodium ion, there is movement of glucose also. It is called an example of co-transport. You might have seen some revolving doors present at the entry point of certain park. There is a revolving door. There may be two possibilities. One person is entering from outside to inside. There is a small area in that. Another person also comes and he also tries to enter along with that person. That is an example of co-transport. And there may be another situation where one person is there for entry from outside to inside. There is another person coming from inside, he has to go outside. And at the same time they are utilizing that revolving door. Along with revolving door, one person which is present on the outside moves inside, and the second one which was standing from inside, it goes to outside. So similar situation is there in the counter transport. Here, due to a concentration gradient developed by the primary active transport in sodium ion concentration is high here, less here. So in this carrier protein, what happens? Sodium ions move from outside to inside, and at the same time the calcium ions move from inside to outside. Similarly, in kidney tubules we get an example where sodium ions move from outside to inside, and at the same time hydrogen ions present on the inside, they move to outside. So this is an example of counter transport. So this was about active transport. Now what happens? Recently, in last few years, some recent researches have taken place describing role of Clathrin proteins in endocytosis. Endocytosis is movement of liquid or solid from outside cell to inside. It includes both phagocytosis and pinocytosis. So during last few years, what happened? So this is the Clathrin molecule. Clathrin molecules were discovered in 1970, and they have a tripod-like structures. They have a heavy chain of polypeptide and a light chain of polypeptide. Three molecules of Clathrin attach with each other to form a tripod-like structure, and again, like the covering of a football or volleyball, these triskelions attach to make pentagons or hexagons. The number of pentagons may be more than 12. So these Clathrin molecules make a cage-like structure, and during transfer of molecules from outer surface of the cell to the inside of the cell during endocytosis, just after formation of the vesicles, these Clathrin molecules attach with some adapter proteins and ligands, make a cage-like structure around the vesicles. And then after some time it detaches, enters into the cytoplasm. In the cytoplasm, the cage-like molecules again detach. They again go to the cell surface for utilization once again, and the vesicle then fuses with endosome to form lysosome. So this model was proposed by Barth D. Grant and Miyuki Sato in 2006. They have written a very good review article in a book, Worm Book, and they had performed their experiments with nematode Caenorhabditis. So here you can see that there are Clathrin proteins. At the cell surface we have many receptors and ligands. So these receptors and ligands bind with adapter protein, and with this adapter protein the Clathrin molecules bind. This binding makes a sinking of the cell surface like this. A vesicle is formed, and around these vesicles these three molecules attach and make a cage-like structure. So the formation of Clathrin coated vesicle. When this cage-like structure is formed by the action of GTPase dynamine, there is a just pinching off and it detaches. It comes into the cytoplasm. Then by the action of some molecules, chaperone HSC70 and DNA J domain co-chaperone oxidine, these cage-like molecules formed by the Clathrin, they just detach, and Clathrin go near the cell surface for reutilization once again. Now these vesicles bind with a endosome to form a primary lysosome, and in these lysosomes further treatment of the foreign molecule takes place. In oocyte formation of Caenorhabditis elegans, Barth and Miyuki Sato have also suggested a model, yolk protein 170. It is formed in the intestine, then it is secreted into the body cavity. From the body cavity, on the cell surface of the oocytes, there are certain yolk receptors called RME2, and these yolk receptors bind with the secreted molecule, yolk protein, and then transfer the yolk granules inside the cytoplasm of the oocyte, and then the yolk receptor again comes near the surface. So these were some new researches in the field of intracellular trafficking. Thank you very much.